So for instance, we know that root mycorrhizae interact with their host on multiple spatial scales, and that the soil that they are interacting in can change over the scale of centimeters to meters to kilometers. And so that begs the question of what's important in terms of shaping that interaction across those spatial scales. So the system I work in today faces a lot of variation as well, and this is the Anthoplura system. And I'm going to give you a brief introduction to the host and the symbiont, and then sort of results thus far about what's happening across their ranges. So my three species that I work with are all in the genus Anthoplura. So there's Anthoplura elegantissima, which is a small clonal polyp that forms these really large aggregations in the intertidal zone. And then two solitary polyps, Anthoplura sola and Anthoplura xanthogrammica. Those tend to be found lower in the intertidal zone. They're large and solitary, and don't form those large aggregations like elegantissima does. And although these organisms are sessile, they have an interesting life history that allows them to disperse really long distances. So the adults are sessile, like I said, in the intertidal zone. They release their gametes into the water. Those gametes then join together to form planktonic planula larvae that look remarkably similar to my illustration. They can be in the water column for weeks, so they can go anywhere from 3 to 4 weeks, it's estimated, and that gives them a really long time to potentially disperse very long distances along the coast. So they're not necessarily going to end up where mom and dad started, and that can be really important for them, especially if they start somewhere like Washington and then end up in Central California. Those two places are very different, just in terms of broad sort of environmental variation. They then settle out of the water column where they are juveniles. They might move a little bit after that, but not very much. And then they're basically sessile the rest of their life and have to deal with the conditions that they basically are stuck with. So the other part of this system is Symbiodinium. And so this is the same symbiont that's found in corals. In this case, it's found in all three species of Anthoplura, and if you took a cross-section of a tentacle and looked sort of in that deepest tissue layer, you'd find a bunch of single-celled organisms that look really similar to that. And they're photosynthetic, so they're providing the host with sugars, and in exchange they get a nice place to live. And so that's the same relationship that forms the foundation of coral reefs, and a lot of what we know about Symbiodinium and what they do comes from coral reef environments. And so, for instance, we know that there's a lot of diversity within the Symbiodinium lineage. So this is broadly all of the Symbiodinium that we've characterized so far based on a few loci. And they group out into these sort of large clades that we characterize them by. And these clades are known to have different sort of properties in terms of the environment that they like to live in. And we see, for instance, that if corals bleach, they'll lose a certain clade and might regain another clade that might help them in that time when they are more stressed out, or perhaps they're parasitic. It's sometimes unclear. And we also know that over the span of like a single coral head, that this brighter environment that might be found here might favor a symbiont host that's different from the one that's found underneath it. So there's sort of these environmental scales over large distances and small distances that matter for the relationship between the two. So Anthoplura on our coast are unique in that they only host one clade of Symbiodinium. So they all host clade B. And so any variation in terms of what the symbiont is doing for the host is going to be found within that clade. And up until recently, we haven't really been able to tease that apart because when we genotype them, they've all just fallen into this clade B, and we don't know about the variation that underlies potentially what they're able to do in terms of providing the host with sugars. And so just to give you a sample of where these guys live, so if you're in the intertidal zone and you land and settle out, say, on top of this rock, you'll face a drastically different environment than one you might face just a meter below you, right? So up here you'll face sort of this hot, dry environment where once a day you might be left exposed to the heat and sun for multiple hours at a time. Whereas had you settled or moved just below there, you might never be exposed to the air at all. You might be shaded, and that scale of variation is just over a meter or so. Likewise with elegantissima, since it's clonal, the aggregation itself might face different environments depending on where you are in that clonal aggregation. So you might be on top of this rock and exposed to the air. Meanwhile, a genetically identical individual to you is basically submerged all the time when the tide is low. And just to give you a sense of scale, that group belongs to my PI, Grossberg, and these aggregations can get quite large. So they're potentially square meters with thousands of individuals. And you can see that they span sort of multiple environmental ranges from sort of this shaded environment to maybe being up here at the top. We know that being on the outside of an aggregation versus inside the aggregation changes your thermal environment, for instance. And so where you are might matter a lot for that symbiosis and what it's able to do for you. So the sort of broad questions that I'm going to address today is looking at whether or not there's genetic structure in the Pacific coast, in the host or the symbiont populations that might allow for specialization of either or both on regional environmental scales. So looking sort of at the biggest large-scale picture of these guys, how might they change along their range? And then asking the additional question of whether or not symbionts specialize on host species. And if so, looking at alternative hypotheses about whether or not they specialize on host environments. So for instance, maybe symbionts in those large solitary polyps tend to look more similar because those large solitary polyps are providing a similar environment. Or maybe they're just specializing on the lineage and there's no actual indication that the environment the host is providing matters to them. Brief methodology of what I did. I collected the tentacles from all three species in 8 locations along the Pacific coast. I genotyped them using RADseq, and then I first aligned the reads from the genotyping run to a draft elegantissima genome that was provided from D. Denver's lab at Oregon State. And from that, I am able to call host loci relatively easily. I then take the remaining reads that don't align to that genome and do a de novo assembly using Jonathan Piritz's DOSIN pipeline. I take those reads then and blast them to known symbioidinium genomes. So there have been 2 published, and I blast to those. Anything that does not match the symbiont genome or alternatively matches bacteria or other cnidarians, I exclude from the data set. And then I'm left with a set of loci that I call symbiont loci. So I'll start with the hosts. And specifically, I'm going to talk about elegantissima because I have the most data collected for them. So that's the small clonal polyps that form large aggregations on the coast. If we go down the coast, what we see is that starting up in northern Washington down to southern California, that these hosts are not panmictic despite that long larval duration that they have, right? So despite that potential where they might disperse long distances, what we see is that there's genetic structure along the coastline. So this is an admixture plot where each horizontal bar represents a single individual, and what admixture tries to do is predict the proportion of their genome that is made up of, in this case, each of 3 populations. And so what we see is that there's sort of this northern blue population that tends to fall out, and the southern population in La Jolla that's green. And then we have this sort of isolated Big Sur population. And if you run tests on this looking for sort of demographic parameters using the program DADDY, you find sort of what you might expect given this admixture plot, that there are relatively high rates of gene flow between this northern population and the southern population relative to gene flow into this Big Sur population. So this Big Sur population appears to be sort of oddly isolated from either the north or the south populations. It's sort of unclear why, but this structure might open the door for these hosts to specialize at least more broadly on sort of environmental variation that they might encounter along the coastline. So if we now turn our attention to the symbionts, I'm now incorporating data from all 3 host species into this graph. And so elegantissima are the same individuals that you saw in the previous plot. They're all shown in red. And additionally, I brought in genotypes from Xanthogrammica from these 2 populations on the central California coast, and then genotypes from Sola from these 3 populations on the southern California coast. And what we find is that broadly, the symbionts appear to be segregating by host species. So there's some variation within the clade B symbioidinium on our coast, and it appears that they partition themselves based on the host, the species that they're interacting with. And what's notable here is that Xanthogrammica and Sola are 2 sort of solitary species that live in roughly the same place in the intertidal zone. They appear to be hosting a different group of symbionts from each other, which implies that sort of their gross morphology is not determining the symbiont community that's living inside them. And elegantissima sort of has its own clade here. If you run this through structure or another Bayesian technique to look for structure broadly across these, it pulls out Xanthogrammica, but not necessarily Sola from elegantissima. So it groups these 2, and then Xanthogrammica sort of stands alone. And so what this basically tells us is that the hosts are not genetically homogeneous across their range despite that long pelagic larval duration. So we can't just take a pelagic larval duration as a surrogate for the potential dispersal distance that the host might enjoy. They potentially are quite limited in where they're going, and over sort of broad spatial scales, it looks like they're genetically differentiated, which implies that they're not exchanging on migrants. The symbionts appear to specialize on the host species, which is something that we didn't know before, so we had just called them clade B for a long time, but it appears that that specialization is there and is apparent in that PCA plot. So that means that, say, if Xanthogrammica experiences some change in its environment, its interaction with its subset of symbionts might be different from the interaction that happens when Sola experiences the same change, right? There are two different groups of hosts and symbionts that might be interacting here. And then the next steps and things that I'm currently working on are looking for more fine-scale genetic structure within the intertidal zone. So if you're, say, high in the intertidal zone versus low in the intertidal zone, can we partition the symbionts across that environmental gradient over very small spatial scales? Can we partition the hosts over small spatial scales as well? That's stuff that I'm working on that I look forward to presenting to you in the future. So with that, I'd like to acknowledge the following people and undergrad assistants and funding agencies. And I'd be happy to take any questions. Historically, people have found genetic breaks across point conception, which spans that point. I'm not sure if that's what's driving it in this case. That's my best guess. And yeah, Big Sur is sort of my big mystery that I have no idea about. Yeah. It sort of plays out in the symbionts too. I didn't point that out in the Elegantissima, but the southern genotypes tend to partition more in Elegantissima more for those two populations. And I don't know if that's part of it too.
How's the transition? Do they come up once they settle, or?
Yeah, horizontally, probably near settlement, if not after.
What's the FST between these groups? Like, how diverged are the red, blue, and green clusters?
Yeah, the question is how diverged are the groups of hosts, and they, so Elegantissima has FSTs anywhere from like 0.03 to 0.12 along that range, but there are actually populations of Sola and Elegantissima that are more closely related. So I can find smaller FSTs between some Sola and Elegantissima than I can from really distant Elegantissima populations. Yeah, I also can't, none of my SNPs are like definitive for Sola. Like I can't find anything that's diagnostic